CSU. On behalf of the Education Committee, I'm Neha, and we are hosting actually Dr. Amanda Hulse Kemp. She is going to kickstart our webinar series with her area of expertise in bioinformatics. And we have Daniel here who's going to introduce her to us. Go ahead.
So, hello everybody. My name is Daniel. I'm a postdoctoral researcher working with Dr. Amanda Hulse Kemp. I'm a bioinformatician and I've been working here for about 4 months. So, Dr. Amanda Hulse Kemp, it's a bioinformatician leading different strategies to analyze she's working with different cultivars and different species, and yeah, she works at the USDA-AGRICULTURAL and also as a professor at NCSU.
Okay, well, thanks, guys. Neha, can you see this popping up?
No, I do not see it popping up.
Okay. Okay, so today, hopefully, we can go over a few things to learn about where we are in bioinformatics with plant breeding. So I've just tried to do a lot of assessment of the data that's out there and to try to give you guys an overview of what we're looking like going into the future. So to just give you a background, I do work for USDA-AGRICULTURAL RESEARch SERVICE. So I like to put this at the beginning so that everybody knows a little bit more about ARS. So we're interested in finding solutions and improvements to agricultural problems that affect Americans every day from the field to the table. So you may not know this if you're at a university location and not working with ARS, but there are actually 690 research projects, and those are split up kind of into 15 national programs, which would be like departments at your university. There are about 2,000 research scientists, PhD level scientists that work with USDA, and there are 90+ research locations. So I work with the Genomics and Bioinformatics Research Unit that's led by Brian Scheffler, and our unit is hosted out of Stoneville, Mississippi, which you can see on this location here in Mississippi on the left part in the Delta. So what we're interested in our particular unit is generating a lot of data and analyzing that data, and then the integration of that data into field programs. So we are located in the southeast area, which I've shown there on the right. And me, physically, I am located in Raleigh here at this purple star. And our unit is kind of special since we're bioinformaticians. We can be spread out and work at different locations to work across the southeast area. So there are people in my unit, scientists, at each one of these star locations. So moving forward, I asked most of you guys to do a survey. So it appears that most people have actually heard of bioinformatics. We're kind of evenly distributed, having graduate students, industry, and public researchers on with us today on the call. But the problem is that even though most people have heard of it, most people would say that they're not really familiar with it. And if you remember our options, the average for saying where you work and how you've heard of bioinformatics, most people are actually skewed to people that have not worked with it themselves. So that means we can make the assumption that a lot of us are pure plant breeders and spend most of the time in the field. I'm hoping that that's the case based on the survey. And those that have heard of it, I asked everybody to define what their definition of bioinformatics was and generated this word cloud. So if you're not familiar with word clouds, it's basically taking the more abundant words and making them larger into the cloud. And I tried to make a tree in this particular case. That's for plant, and that was the only plant option. But we can basically see here our general consensus of what our definition appears to be is that bioinformatics is using data analysis tools to assess biological data. So that's, I would say, kind of going in the correct direction, but perhaps it's still a little bit not known what bioinformatics is. So today I really want to talk to you about what bioinformatics is, how it has been used in plant breeding thus far, and if you wanted to get into bioinformatics, where can you go to get some resources to get started? And if you need help, then where can you go? and then really looking to the future, what promise does it hold in the future, and how can we use bioinformatics in plant breeding. So just to start with, what is bioinformatics? The first known use, according to Merriam-Webster, was actually in 1975. However, a lot of people go back to bioinformatics as starting being coined by Pauline, who is shown here on the right, in the bottom right, in 1970. So that was in a pretty hard to read paper and hard to access paper because it was written in Dutch. And of course, most science articles are in English, so that's hard for most of us to access. But here is the depiction in 1978, and she actually is working in a system where you can see that she is now coined in an actual paper in 1978 the word bioinformatics, and claiming that to be her main field of research. So interestingly enough, she has another paper here more recently, and you can see that she's still working in bioinformatics, and I think that that's as recent as 2011, so maybe the person that coined it actually is still there working in, I believe, the Netherlands. So in 1985, the Bioinformatics Research Journal was actually published, and you can see the current cover of that down at the bottom. So we've had bioinformatics actually for quite a long time. I guess we're coming on 50 years, I suppose, with technically bioinformatics. And just thinking about it, when we think of our field of science, we of course have kind of these individual fields. So you could get a PhD in molecular biology, a PhD in biology, a PhD in computer science, or a PhD in engineering. But oftentimes we actually have these kind of middle of the field overlaps. And that's kind of what we're talking about when we think about bioinformatics. So bioinformatics, in my opinion, is kind of the between the boundaries between biology and computer science. So maybe that overlap area in the middle there. And a lot of people actually have a couple different definitions for people that work in bioinformatics. And my theoretical understanding of this is that you can come at this from both sides. So you can either be a computer scientist that starts to learn some biology, and in that case, the technical definition would kind of be a bioinformaticist. But you can also be more of a biologist and come at bioinformatics from the biology side. So those people, I would say, are computational biologists. So for me personally, I would put myself on the left side of this figure. So I have an undergrad degree in biology. My PhD is in genetics. So I would say that I am a computational biologist rather than the person that comes from computer science and gets into the biology side of things. So there's kind of a mixed mash of people that are doing bioinformatics, and the real technical, I guess, split between these two people is that the computer scientists are more able to write code. So they're the ones that are working on software development, making algorithms faster, putting together large databases, looking at that type of thing, whereas the computational biologist is really trying to take those tools the bioinformaticists develop and make efficient platforms for using them. So that's my definition for somebody as to what the difference between those things are and where those terms come from. So I'm just going to continue along this thought throughout this call as really the view from a computational biologist standpoint, not so much a bioinformaticist. So the first thing that we want to talk about is actually the data production in science. So sequencing really was the first intensive level of data generation. So we can see on the right here kind of a timeline of NCBI. So in 1988, the National Center for Biotechnology Information, as we all know as NCBI, for storing sequence data was developed. So we can see from the beginning that there was pretty much no data to at the point where we had tons and tons and tons of data. So this only goes to 2015, but I'm sure that we're still on the exponential growth curve for generating data, and I'm not sure that we'll necessarily ever get off the exponential curve. So that kind of plays a role in what we need bioinformatics to do, because we're generating so much data that without informatics tools, we can't actually process through that data anymore. So the first bioinformatics tool, I suppose, that was widely implemented and most people still use it today is you can think of BLAST, which is on the starting of this time curve here. So if you want to sequence or assay something and find a sequence in the NCBI database, you still go to their BLAST server. And you search their whole database of all of these tons of sequences through that algorithm. Yeah, BLAST is a free. So Neha was asking if BLAST is free, and yes, it's a free thing. If you go to NCBI and just Google BLAST, you'll find the NCBI link to use BLAST to search their database. So I was really interested in kind of the increase in bioinformatics publications over time. So just taking that 1970 as the initial start and coining of the term bioinformatics and looking at the publications over time to current day. So you can see pretty much not until 2000 did we actually start seeing any bioinformatics publications, and then it kind of increased after the year 2000. So the significance of that really is we could go back to our sequence time curve, and we can see that the human genome was finished in 1999, and at that particular point in time was when we really started generating a lot of data, and when it became necessary to start using informatics tools to analyze the sequence data. So that, in my mind, kind of makes sense why we don't really see anything until 2000, because if we think about it, that is when we started sequencing plants as well. So here I've added plant breeding to the curve. So I'm finding all of this information, basically going to PubMed and requiring these search terms in my search field, and just counting the number of publications, which you can see on the y-axis across years. So compared to bioinformatics and computational biology, even though plant breeding has been around for a very long time, we basically are kind of at the nothing level compared to the number of papers in bioinformatics and computational biology, which is a little sad as somebody that comes out of a plant breeding background, that we really have just a few papers that we're able to find, but perhaps that's because we do a lot of things that are not really translating into publications. And then moving forward from that, I'm actually interested in a lot of those plant breeding papers actually involve bioinformatics and computational biology. So I searched for both plant breeding and bioinformatics and plant breeding and computational biology, which are the two lines that are basically on the zero down there below everything else. So just zooming into what is actually here, so you can see the levels of publications each year are around 30,000 and 20,000 for bioinformatics and computational biology in the last couple of years, but plant breeding is down towards less than 3,500. And those plant breeding articles, the ones that actually involve informatics, are down closer to maybe 200 per year, which is pretty interesting since we know we have a lot of students and people actually working in these fields now. So that brought a lot of questions to my mind. Well, what's actually in those publications, and what can we learn from that moving forward? How can we get more bioinformatics into plant breeding to help speed up our plant breeding process? And this is just looking at only those plant breeding intersections with the informatics. So I think it always tends to, bioinformatics is a little more hot keywords in computational biology. So I think in most of these graphs, bioinformatics tends to be at the top. So you can see there's about 100 more papers for bioinformatics than there is for computational biology. And since we actually started using it in plant breeding, there's about 2 decades worth of papers. So I just made the arbitrary split here, which places our stuff into decade 1 and decade 2. And I'm going to look at what's actually in these papers. So I'm generating a word cloud again. That's the titles of these papers. So you can see on the left the word cloud for 126 papers, which was the first decade. And on the right, there's over 2,000 papers for the second decade. So we can see that in the first picture, it appears that there's not really too much of a general consensus on what people are actually doing in those papers to pull them out. There's no theme really, aside from genomics and plant analysis and comparative analysis. But on the right, then we can see that we've got some pretty definite themes there. We have a lot of big words that are dominating this, And we can see some big crops there. So we can see cotton, rice, maize, and then of course genomic selection and genes and crops. So kind of, I guess, what we would more expect to be happening. So taking a further look into that first decade of material, we can see that the first bioinformatics in plant breeding, there's only 4 of those papers that are actually mentioning selection. And most of the ones in plant breeding are actually all review and opinion articles. They're not actually applying bioinformatics, they're talking about how they might possibly be able to do it in the future. So they're really focusing on the utilization of genomics, which follows DNA sequencing being the first kind of big generation of data. So the first paper here was Genomics in Plant Breeding. This was in 2001. And the big point, highlight take-home point of this paper was that we really need to create an ESP database. And I think if we think through what NCBI has now, we really have a pretty good ESP database for most of the crop systems. We can kind of check the box on what this person was thinking we needed to do for the future. And then the second one was really talking about plant genomics and breeding practices. So this was a nice figure out of the paper that you can see that bioinformatics is actually there for mRNA profiling and protein profiling. So now we're starting to talk about the different omics. So of course we have genomics, which showed up in our word cloud, transcriptomics, proteomics, metabolomics, they're all showing up in this graph. So we can think about moving from phenotypes and phenotypic variation, looking at the genes that are controlling those, and then moving those into plants that we're interested in selecting in our breeding process. So this person in 2003 was actually hypothesizing that we can stick bioinformatics in kind of all of these different steps here to understand the biology, to get it out into plants that we're breeding to put in the field. So rice was the first crop plant sequence, and interestingly, the bioinformatics in this paper really were kind of limited to what was available, and that just happens to be Arabidopsis in humans. So the bioinformatics in this paper were really for designing the capacity for putting together genomes. So the first decade really focused on bioinformatics tools in order to put together genomes. So I would say that that's what the first set of papers had in common. So you can see the figure from this paper on the left is basically picking out how to mark repeats, how to deal with repeats into short sequence data, closing those gaps and trying to put scaffolds together for sequencing. So scaffolds are these little pieces of sequence that we're able to develop with short reads, and we basically want to get those to a chromosome length assembly so that we can actually start using that to localize things of interest. So once we're able to localize the things of interest, then we can follow genes as we're breeding our plants to get something of interest in the last selected set of materials. So the one thing they did was they looked at the Homo sapiens intron exon distribution they compared the rice to Arabidopsis and to human. And we can obviously see that human is much different than the plant. So this kind of told us that we need to, of course, stop comparing to humans because the plant is nothing like humans, but to start generating more data for plants and developing that ESP database so that we can actually look at what's going on in plants. They were also working on the SNP detection as well and started to look at the function of these genes. So this was really the first step into whole genome look. So rice was the first crop plant that was sequenced. And we put together a timeline kind of on the analysis of genomes. So of course, the first one sequenced, everyone's pretty familiar that Arabidopsis was sequenced right after the human in 2000. Rice was the first crop plant in 2002. And then it wasn't until 2006 that the next crop plant came out, and that's grape. So our trend here appeared to be that we were able to sequence small diploid genomes. So that's what we can see is everything through tomato is diploid. So we were really limited to diploid in our technologies here until 2012. And then the first one that I've shown here is cotton in 2015. And then most recently, we were able to actually sequence a lot of polyploids. So everything here at the end is actually coming back and sequencing our important crop species that happen to be polyploids. So peanut, wheat, sugarcane. And sugarcane, of course, is only a haploid representative for what we have now. And by 2013, we actually had over 50 plants sequenced. And at this particular point in time, we now have most major row crops sequenced. And now we're coming back and working on some horticulture, ornamentals, a lot of the smaller crop species that didn't really have the money that these major crop species had in order to do genome sequencing. So again, if we think about our distribution of the 2 decades of doing bioinformatics, the first plant in the second decade would really be soybean. And we only had a few genomes that were completed in that first decade. So we're kind of limited on the genome sequences at the first decade of papers, and we're really building up our sequences by the time we get through our second decade of sequences. So thinking about the first genome in rice, then we can start to think out where have we actually made it when we get to the last genome that was sequenced. So peanut just recently came out last month in Nature Genetics, and peanut, of course, is an allotetraploid. So as I mentioned, that's kind of where we're getting to now, is that we're sequencing a lot of these polyploids. So previously, the mind of the informatics person was that we could utilize the diploid ancestors of the tetraploids and higher polyploids in order to try to get this information out. So technologies have actually gotten better, and we sequence on long-range sequencing read technologies like Pacific Biosciences and Oxford Nanopore to get these long reads that allow us to actually separate subgenomes. So that makes sequencing much easier for polyploids, at least in the allos. In the autos, it's still pretty difficult, so those would mostly go onto the list that haven't been sequenced yet. So there's a lot of people still working on that. But this last peanut, we're able to actually start to understand evolutionary origins of these polyploids. So for peanut, they found that there's a single polyploid origin, and they were able to sequence most of the plants on the right that had some impact on the evolution of cultivated peanut. So cultivated peanut is actually shown here in V, and this is just a representation of their chromosomes and the different genes from both subgenomes mapped back. So they're interested in looking at homologous recombination here and what happens. And we can see that both tip runner, which is the main cultivated species and the one that was used to develop the sequence, that it behaves similar to this other variety here. So we can see at this particular spot on chromosome 5 that there is actually a complete representation of those from the A genome rather than from one copy from the B and one copy from the A, which is what the majority of the genome is showing. So they were able to detect this region of homologous recombination with this level of bioinformatics, with this new technologies that are available now. So we're starting to see what's going on with homologous recombination. You can start to look at recombination events and this type of stuff with the better data that we have now compared to the last decade of bioinformatics. So our bioinformatics cloud that we're looking at for our papers in the second decade up there, remind ourselves on the right, is that we now have many crop genomes. And with those many crop genomes, we can actually look at determining the basal ancestor of crops. So the first instance of this was actually in grape, so quite a while ago, but we've been able to refine this based on the number of genomes that we have. And we can now identify the U-dicot ancestor of crops actually have 7 chromosomes, and those were most likely triplicated, which are shown at the second level here. And we're able to look at all of our crop species and say, well, what happened throughout the evolution of time and where do these genes go? So this is kind of a very nice example in eucalyptus. And you can see that if you look according to the different colors of the ancestor chromosomes, the eucalyptus has this really nice blue one. has this really nice blue one that is basically uninterrupted compared to any of these others. So it almost has a complete copy of one ancestral chromosome intact. So now we can start to answer questions with bioinformatics, like why is it that eucalyptus has this one particular chromosome intact from many, many million years ago, and others don't. You can see, of course, in all of these others that most of them happen to have complete rearrangement of this eudicot ancestor chromosome. Grape has probably the least amount, and that's because they found that it has not moved around as much as why they initially identified it in grape and have translated it to other species. So if we look at our word cloud and the words that are starting to pop out at us, it also tells us that we started to begin looking at the wild relatives, and we're able to use large data sets to start to reveal gene function. And one example of this that I guess happy that it popped out is in cotton, since my background is actually working in cotton. So there's a really nice paper that recently came out where they're doing a lot of work in this data, and that we're now looking at, instead of a single representative of species, many, many representatives of species through bioinformatics. So now we can actually look at hundreds of lines of the same species to try to figure out what is causing certain phenotypes. So this particular phenotype that's shown here is Fusarium. And they were able to run a genome-wide association analysis in A and pick out SNPs, so single nucleotide polymorphisms that were different between those 243 lines, and find a place in the genome to look for a gene that would be responsible for this phenotype of interest. So they found this gene here that they were then able to transform, and you can see that the uninoculated and controlled plants did not get Fusarium, but the ones that they altered did. So they were able to use this information to prove this phenotype, as many, and I think there was maybe 3 other examples in this particular paper to actually prove what specific changes in genes are actually doing by using large data sets. So you can imagine the resequencing of 243 lines is probably generating lots and lots and lots of data. So you need bioinformatics to try to answer these problems. So the other thing that kind of stood out to me is that we had genomic and selection in our picture up there. So genomic is, of course, the biggest word occurring the most time, and then selection there. So I took a look at what was going on in the publications that were actually including selection in the title. So we can see that while it didn't exist in the first decade, that now we're kind of seeing an increased number of articles associated with informatics that are looking at selection. So we can see an increase over time, especially recently, in the number of articles that are including selection. So these primarily are looking at genomic selection. This is a nice paper that just came out from the Cornell group, and they put this figure together that kind of nicely shows what has been happening in plant breeding and kind of segregating these 4 different periods of plant breeding. So just looking at their figure, we can see that there's section 1 is really what has happened before 1900. So this is focusing on unconscious selection, people selecting for things, but not really in a conscious manner, and then trying to adapt things to certain locations, but not really having any information to do that. They're just picking the ones that look good, keeping the seed from those, and kind of moving forward. So just unintentional, without knowing exactly what they're doing, but still selecting based on what normal humans would do if you had no information. So then once we had Gregor Mendel and his information, and we started learning about genetics in the earlier 1900s, then that's what we're classifying as section 2. So those are really where we can start to control genetics and look for what's going on, but not really having any essential sequence information about that's going on. So we can use, of course, Mendel's crosses to see about what our segregation ratio is, look for heterozygous types, homozygous types, and select for those to move things in one direction. So kind of in a more intelligent manner than what we were doing, of course, back here. And then starting to use some pedigree selection by crossing ones that we like by ones that we also like. So kind of trying to bias what we're doing and moving things in a certain direction, but not really being able to look at the genetic level at those. And then in the 1980s, then we're now moving on to understanding genetics, being able to look at sequencing that and looking at what the DNA markers are at those particular loci. So using marker-assisted selection along with pedigree and Mendelian crosses to select towards things of interest. So these are all involving genetic markers and loci that are tagged with some type of genetic information. And what's interesting is this starts or is coming in really at the point where kind of bioinformatics overlays. So if we think about bioinformatics as starting at 1970 to 75 into the 80s, then that's kind of sitting on top of where these markers, the selections come in. So at this point now, bioinformatics is really kind of coming in to consistency with both 3 and 4. So 4 is really looking at the next generation of informatics, and that's looking at transformation and genome editing. So how can we use informatics to predict and design something rather than just to select for it? So this is using other levels of technological tools on top of the informatics, but using informatics to predict those things. So this is a really good paper that I suggest people to read and kind of wrap their heads around and think about where you are in your system. So we're, of course, all at different levels in our different plant breeding systems. So if we think about kind of outlining our own plant breeding program, everybody has their own outline. Your program is going to be very different from my program, who's different from the person down the street breeding something different. So just think about when you start your program to kind of have this written down to understand what you're doing, but also to be able to tell somebody what you're doing. So a picture is worth 1,000 words. If you can put your program into a picture, then it's very easy for somebody to understand what you're doing. So I've just put a few different pictures of different schemes here. So most of the schemes, of course, as plant breeders, we'll notice that we need to cross something, we need to select it for something, and then we make more crosses, and we do field trials. So essentially, that's pretty much what all of these pictures are showing, and we know that we can do most of those things in a lot of different schemes. So all of our schemes are going to be different, but essentially we're interested in selecting things, selecting them faster and more accurately, picking the best ones, and then sending those best ones out for trial and final selection to get them down to cultivars. So let's just think about that as we're moving along. So I think genomic selection really is probably the best application of bioinformatics in plant breeding thus far, and certainly compared to our word graph, the most well used and most applied at this particular stage in time. So I'm going to look at some utilization of genomic selection and a good application, because I think that this is the stage at which most programs are sitting, where bioinformatics is actually utilized most in order to get data into and analyzed for genomic selection within their breeding program. So you need bioinformatics really to obtain the inputs and then analyze the data for genomic selection. So if you're doing genomic selection and you don't think that you know what bioinformatics is, I hope now that you recognize that maybe you're some level of bioinformaticist or a computational biologist if you're doing genomic selection or running genome-wide association models. So anything really where you're looking at genome scale data, you need informatics in order to analyze that and get that down to something that you could use in your plant breeding program. So thank you again to Jim Holland for letting us use this as an example. So just looking at a program of genomic selection, we can think of we kind of have bioinformatics input from two sides at this point. So we have the informatics that are coming for inputting our sequence data So most people are focusing on sequence data into these models now, and that's largely due to the cost per genome is going down. So we can get a lot more sequence data for our money, but that also means if it costs less, then we're probably generating more. So we're generating more data to get sequence data to put into our model, which is our bottom half of the bioinformatics here. And most people have been using the Tassel software, which was developed at Cornell, maybe with the technology genotyping by sequencing or resequencing, sequencing of some manner in order to get these SNP data for individuals to put into a selection pipeline. And many people are also using custom pipelines here to get their genotyping data. So on the other side, we're actually also using bioinformatics for high throughput phenotyping. So there are a lot of people that are looking at using drone data, which is shown on the left here for, I assume that this is probably NDVI data. So normalized vegetative index. So obtaining things like this, ground-based systems for selecting different types of sensors, image-based systems for collecting different assaying things about the plant. In this case, maybe it is plant leaf area that are getting put into these models for genomic selection, and then analyzing those with our statistical software to get to our estimated breeding values that are actually going into our selection pipeline and our breeding programs. So this is just an example of looking at in an actual program 2 different ways of looking at materials. So of course we have our traditional trait data on the left. So this would be using 20 lines on each side. And then on the right-hand side we're actually starting with the trait, but then continuing only using marker data. So this is solely genomic selection on the right-hand side and traditional phenotypic selection on the left-hand side. And then doing field to head-to-head field evaluations in the field to determine which one's actually making the best progress. So on the left-hand side, we start with our 20 lines, we intermate them, and then do a generation of selfing. Once we have that generation, then we're able to do phenotypic evaluations on about 12,000 ears. Once we pick our selected material out of there, then we're going to do intermating again of our progeny selection, and then self those to get to something that we're putting into the field. On the right, then we're running multiple intermating generations in both the normal field cycle in the summer and then sending to a winter nursery in the winter. So we're able to get more generations of crossing here on the right with genomic selection than we are on the left. And then also doing the same number of selfing generations to get to what we're evaluating in the field. So this is going into the field for evaluation this summer, but we can already look at the genetics of this data. So on the left, we can see the original population, which includes the training sample, and these ones were where the parents were selected from. So we can see the parents are selected from these dots, black dots, and then this is actually the genomic selection population. So we can already see that the allele frequencies have been moved significantly, and the distribution of these individuals, the genotypic similarity has really changed in that new population. So we're really getting good selection through genomic selection, and it's changing your population very fast. So things like this and integrating bioinformatics can really get you somewhere quickly, and we're waiting on the analysis of this in field data this year to see if this change in allele frequency also corresponds to significant increases in different phenotypes of interest. So that will be interesting to look at. However, it's of course good to keep in mind that as you're doing genomic selection, that you need to make sure that your training sets are continuing to reflect your new population, so you can quickly break down your models over time. So we really don't want to fall off the cliff with our training population. So I'm going to shift gears a little bit out of genomic selection and to think about where you want to go. So this is kind of like bioinformatics in the big general scheme of things. So if you want to do any type of analysis on big data, that's what we're thinking about in bioinformatics. So what you really need to do at the basal level is to understand basic levels of coding. So there is a nice group put together called Data Carpentry that really does some nice introductory methods for using Linux, Unix, Perl, and Python. So these are all different coding languages. So if you're going to work in bioinformatics, then you need to be really familiar with how these computers work and how to talk to the computers to analyze your data. So I really suggest making sure that you start there. And you might look for a data carpentry lesson class at a location near you. So you can just search data carpentry and find that information. And then perhaps the best way to get started is to actually use these graphic user interface tools. So Galaxy is a free one that I hope many of you have heard of. So the picture of that is the biggest one up here to the right. So you can access that here. And then there are other options for graphic user interfaces but they do cost money. So that's, of course, what the dollar signs are talking about here. But these are two other options with VLC, Genomic Workbench, and Genius. The nice thing about these is that you can actually see kind of a process of what you're trying to do. It's written, you can click on it, you can see what the software generally is trying to do. But the bad thing about these is that they're really limited to only small data sets. This is really only when you're learning. So it's an okay place to start learning, but I would not suggest try to analyze 200 lines in here. It's not going to work. So you really need to make sure that you're paying attention here and that you're using this in order to learn to expand what you're trying to do. So if you're going from maybe 2 lines would work here, but if you're trying to run a full system plant breeding program, this is not the place that you want to be. So you need to get yourself to command line. So think about moving to a more advanced Linux system, and really this means learning to use high-performance computing machines, or what people call supercomputers. So on the right there is a picture of one supercomputer, which is found in Texas with the Advanced Computing Center. So this particular computer is associated with CYBERS. So you can see that CYBERS has been working, it used to be iPlant, but they have been working with a federally sponsored programs for setting up learning environments. So you can analyze data, and they actually have some learning tutorials, which you can see down here at the bottom left. And there's a list of the biggest supercomputers. So I guess in the US we can pat our backs that we recently got #1 and #2 back in 2018. Used to be machines in China. So we now have those back. So if you're interested in starting bioinformatics projects, there's a couple different places that you can start. So most of these are, of course, all thinking about coming this from the biology side and assuming as we're plant breeders that that's where we're going to come from. That you can actually look at the Iowa State Bioinformatics workbook. They've been working very hard to put a lot of examples on how to do different pipelines. A lot of it is focusing on sequence analysis, but as I see things moving forward, I hope it will also include many other types of analyses. And the Galaxy graphics user interface that I mentioned before actually has a large number of tutorials that you can use. So that's another good place to look. And then really become familiar with your software. So it's nice to look at the GitHub manuals. So a lot of bioinformaticians that are writing and developing these software do hard work to put these manuals together. So you really need to get to know the manual for the software that you want to use so that you know which options to use. And we call those parameters. So you can see an example of the Bowtie software manual on GitHub at the right here. So it's really telling you what is Bowtie, what's it doing, what are the options, how can I change it. So what you need to know is the biology of your background to be able to change the parameters to reflect the biology you expect to see. So that's the most important thing about bioinformatics, is to keep in mind what the biology is that you're looking for. So make sure that the computer is giving you information out of your analysis that actually makes sense. And you can see that these manuals are actually pretty long. This one in particular goes to line 1700 at the end of the manual. So there's a lot to read there, but you need to read that in order to make sure you know and understand what you're trying to do. If you have problems aside from the software manuals, then there's a lot of people actually that are working on bioinformatics that can answer questions. So Stack Overflow and Seek Answers are really good places that you can go to look, and most people are pretty helpful. If you're working on a high performance computing supercomputer, a lot of those also have kind of question support boards for working on within those. So the group of users that work on those machines are kind of connected, and they can answer questions for within their working group. And then, of course, there's always Google. It's maybe the best machine learning even for plant breeders. So don't forget the good old Google. So just coming back to where we are here in the phase of plant breeding. So what I'm really hoping is that we can think in how in this generation 4 we can actually start integrating more and more bioinformatics into our platform. So we can see that at this point, really genomic selection is probably as far, and high throughput just touching the very beginnings of high throughput phenotyping to include into our plant breeding programs. So I really see it as kind of an opportunity of getting in on the ground floor almost at this particular point in time for integrating informatics into plant breeding systems. So we need to start thinking what really does the future look like and how can we get there. So I really see plant breeding moving from a single crop to leveraging a cross crop, and then using that information and informatics really to push us to new adaptive peaks. So if we think about our selection, usually selection always pushes to the most local adaptive peak, but that doesn't mean that it's the best one. So we really need to try to leverage the informatics and information in different species so that we can really find out what is the best peak and how do we get there. So that's one thing. The next thing that I think of is really, we are using genomic selection, but how can we actually get to the point where it's feasible to use it in both of our plant breeding programs? And what we need in order to get there is that we need consistent, accurate, and cheap markers. So the, traditional way to do this has been using array or PCR markers, and we need to get those to a cheaper, targeted point that we can actually use that. And then de novo sequence information is also important, because if we're going to get to that new adaptive peak, we don't need to worry so much about the old information. We need to get to the new information. And perhaps the promise for doing that is actually amplicon-based sequencing to keep the sequencing costs low, but to get that new allele information for our different, better lines. And in order to do that, we really need better integrated systems that we can use in the field, and also really for all crops. So you'll notice in our word cloud there that predominantly it was the big crops that are there in the word cloud. Most of the others have got lost. They're not moving along with us. So there are some efforts now kind of to bring those back along, such as the Breeding Insight Project, which is focused on horticulture crops. And then also some systems such as the breeding API. So that's interested in taking all of these kind of tools that were developed for plant breeders and making a place where bioinformaticians will manage their interface. So that way you can use all of these tools and not have to worry about making them work together. So if someone will make them work together, you would just be able to use them in the plant breeding database. So that is kind of the ultimate goal, to get all this stuff into a place that someone can actually use it. You don't have to worry about trying to manage it, why it doesn't work together. Someone will take care of that. You can just use it. And then we can build new tools and actually integrate those into the process too. So that is kind of where I see that going. But in order to really do that, we need bioinformatics for understanding our history. So you can imagine that at this particular point in time, we are now sitting at the end of a long history of selecting materials to get where we are. And I'm sure once you have your position or get into your position that you will notice that you have a lot of information and you're not really sure how to put it all together. So one of the things that is kind of at the primary front is trying to figure out how all of your material relates to the rest of your material, how it got there, what is the history of your breeding program, so that you can leverage that data. So oftentimes that means going back to people's notebooks, finding data from papers in 1950 or 1900 or lines from the 1700s that you basically have no information on. So So we can think about doing data mining for actually developing and understanding the pedigrees of our lines, and then integrating all of these different networks into what are called knowledge networks. So knowledge network on the left there is actually a flexible representation of a community's data, or you can think about it as your plant breeding program that allows for actually learning the reason for the connection. So this is kind of just an example of something like that Facebook uses to figure out, well, I'm friends with Neha, and Neha just recently went to China. Perhaps Amanda actually wants to go to China. So it can pull all of these different weird kinds of information and actually help figure out what is the connection between these points. So we can think about putting our entire breeding program into the computer and thinking about connecting the history of your crops, all of the phenotypic data that you're collecting at all levels, field management data, weather data, sensor development, tracking samples, designing your crosses. Really at every level of your breeding program, you could think about integrating something into a knowledge network. So you could learn perhaps how the best way is to get to designing your crosses. Maybe you need to change something in your field management data, and you can start to assess this with these types of connections that are made possible through bioinformatics and putting together very large data sets. So I think that that's really kind of where we're going. So I'm sure somebody more intelligent than I has specific ideas on what the best way to move bioinformatics forward. I'm happy to discuss that with questions. But I'd first like to thank, number one, my many mentors that have allowed me to learn all this material. So my research leader currently, Brian Schiffler at USDA-AGRICULTURAL RESEARCH, David Stelly was my PhD advisor at Texas A&M, and Alan Van Dyne was my postdoc advisor at UC Davis. And then my current and former lab members with the Genomics and Bioinformatics Research Unit, and then my lab members here currently, Daniel Ashley Shoemaker, Montana Knight, and of course many, many collaborators. As a bioinformatician, you find that you're working with many different groups and many different funding agencies. So with that, hopefully if you have questions, we can open it up.
We have about 6 minutes for any questions.
So I think that everybody's probably muted, but I think that you can put questions into the Q&A.
Yeah. Yeah, if you can go and type your questions in Q&A, or even
so I think, so we're just discussing here what is going to come up that's outside of sequence data. So I think that at this particular point, sequence data just happens to be the data that we are generating first. But interestingly enough, sequence data is very small in the scheme of things. So you can imagine the amount of data that's in publications, in text, the amount of data that we're generating, of course, the field with high throughput phenotyping. Sequencing is generated on the maybe say 100 G over 3 days. Our phenotyping plots in the field are generating terabytes of data within a half an hour. So the scale of data for these different generation schemes, in the scheme of things that we're thinking about what we've done so far, is very small. So this will mean that we're going to need more and more informatics once we're generating more and more data. So phenotypes actually,
what just happened here?
Is probably on the scheme of things much, much larger exponentially than sequencing data. It's still alive over there.
It went like what?
So there was one question, if someone's still there, can you comment on the rules for pangenomes and also the relative benefits to conventional breeding versus wide cross breeding? So there are some efforts actually to generate pangenomes now, which look at basically capturing a large amount of diversity within the genus. So you can imagine that this could also be cultivated material and wild material. So the amount of difference, of course, is going to differ depending on which crop system you're in. So you can have things like cotton that have very low diversity, even across species, or like maize that have very large amount of data and differences even within the cultivated system. So these will be kind of different roles, I assume, that will be independent for the different crop species. But the benefit of the pangenome really is that you start to capture that de novo information. So you're assuming that you have all of the genic or possible genes that exist in the genus within a reference, basically.
Right.
So you're able to find it. So if you're doing genomics and thinking about your cultivated material, you have some portion of information that doesn't really represent or is not represented by your reference, and that turns into a problem. Because if you can't find it, then you now kind of just associate it into the unknown, which is what most people do. So the pangenome benefit is that you're hoping that you have a representative for most of the diversity that exists in your program. So I would think, depending on which system that you're in, if you're interested in elite material, then you would probably think about having a separate pangenome for your elite versus your wild. So then you could kind of segregate those and maybe have two different representations. So there's also a couple other questions. So where do we stand with vegetable genomic data in the near future? And yes, it is definitely pretty lacking compared to row crops. So I think with the technological advances that are occurring, some of the issues in vegetable, of course, is that it has large genomes, but it doesn't, they don't have the amount of money that we have in row crops to sequence those things. However, there are a lot of advances coming in the technological side of things that are causing sequencing to still get much and much cheaper. And the long-range sequencing actually is really good for these highly heterozygous genomes, which a lot of the vegetables tend to be. So I think that actually is a promise for the future where we can actually capture haplotype data along with reads. So I think that that's probably one of the best things for vegetables. And I think we expect to see a lot of advances in these kind of orphan-ish vegetable horticulture crops in the near future, particularly with the breeding insight program that's going through ARS right now and the associated programs.
You got another one. I don't know why I want to on that. So
another question that we've kind of touched base on is looking at what the role of high throughput phenotyping is in plant breeding programs. So I mentioned, I think that this is going to be another stage of much higher data generation, which is going to require a lot more work and integration, particularly with engineering. So I know here at North Carolina State University, there's been a lot of discussion and bringing the engineering groups and computer sciences groups actually to the table together. So I think that for us in plant breeding is probably one of the targets that we need to focus on and think about how much data we're actually going to be generating and what type of information do they need from our plant breeding programs we need to keep for metadata in order to analyze that type of data. So this goes back to probably we all know what we do for management strategies in the field, but we really never tend to keep that data. So our field trials, we may treat them differently every year. The weather is a little bit different, so perhaps we put on 3 applications of insecticide this year, but maybe the next year we put on 4. So these types of things will turn out to be very important, and I think the discussions on high throughput phenotyping may actually turn into more, I guess, repeatable experiments in the future and actually tracking of more data. So I think the data generation and all schemes of things in a plant breeding program is just going to continue to increase.
What data you get?
And then I think that kind of translates to another need that I didn't necessarily mention was in the training side of things for graduate students. I think it's definitely beneficial for graduate students to at least get some type of experience in bioinformatics. It's great to be able to actually talk to others not that you necessarily need to be doing it. We do have a lot of people that are mentioning that they're collaborating, right, with a bioinformatician. So being able to kind of tell them what you need and what will help you. So I think that that's really what we need to focus on looking to the future, is to sit down and think in our minds, what does the future look like? What would be the best way to get our breeding programs moving forward, and looking at it more as an interdisciplinary group rather than each individual breeding program. So that takes a lot of work, but I think that the downstream benefits for the public side of things will be quite large. Okay, everybody. Well, thank you for joining. And if you have any questions, you can certainly feel free to email me. I think there may be one more question coming in. So someone is also asking, what other general skills would you encourage graduate students to train? That's kind of a difficult one. I think as students are moving forward, you're getting asked to be more and more interdisciplinary, which means that you really have to kind of broaden your skill set. So I think that maybe working on a project where you're kind of working with different people in different fields would be a good idea. I think that the general communication and ability to communicate between these different fields is what's going to benefit you as somebody in a plant breeding program. You certainly need to be able to talk to those 4 fields that I put up at the top, people in molecular biology programs for figuring out what the actual function of genes are and keeping up with the genes that are important in your system for phenotypes of interest. Those associated ones in the biological field, computer scientists, which are certainly usually, it was supposed one of the last people that you would talk to as a plant breeder. But as you can imagine, as these data generating platforms get more and more, it's going to become more and more important to have an association with a computer scientist. And they may or may not have any background in biology. So the current set of computer scientists generally tend to not have much background at all in biology, which makes it very difficult to try to talk to them and figure out how they can help you get through this mass of data that we're generating in plant breeding programs. I think that that is probably critical, and the engineers as well. So how to get the best data from the different sensors, different high throughput phenotyping to get to your phenotype of interest. So I think it's super important for communication skills, and then of course also for leadership skills. So if you're leading a team as a plant breeder to get you to a goal that include all of these different interdisciplinary people, to have the skills to guide them to move forward to benefit your plant breeding program. So I think leadership and communication would definitely be the 2 top general skills, I think, for students to gain. And certainly learn them while you're doing grad school or your postdoc. That's the best times you can get into the field running once you get your initial position. So I think that's probably a good place to stop. So we'll thank everybody for joining today. And if you have any questions, you can email me. You can find me at amanda.hulse-kemp@ars.usda.gov or on LinkedIn. And if you Google me, then I'm sure the Google machine learning can also find me. So thank you, and everybody good luck in the field. Hopefully you're not flooded like some of our fields. Take care.